So we'll move right into our sessions. Our first speaker is Sharon Presnell. Sharon's career has been focused primarily on the use of primary human cells in vitro and in vivo, while she was in academia, mostly at the University of North Carolina, Chapel Hill. She was also at Becton Dickinson, Tejion, Organova, and Samsara Sciences Incorporated. She joins us today from her current post as president of the Amnion Foundation, a not-for-profit focused on birth tissue-derived cells and services. So I'll turn it over to Sharon.
Great. Can you guys hear me okay?
Yes.
Okay. So I am, okay, can you see the slides?
Yes.
Yes. Okay, great, thank you. So I'm going to talk to you guys today about the use of birth tissue, which is a very plentiful and non-controversial resource to cells, many of which can be used in R&D. And we have a few learning objectives today. I want to make sure that everyone understands the numbers around postpartum birth tissue donation and utilization, understand the full spectrum of the useful cells and tools that are available from birth tissue, and then understand how the culture environment that we place these cells into can significantly alter the answers we get when we use them. And all that is important as we think about translation and alternatives to animals in studies like these. So I like this figure, even though it's beginning to get a little dated, but it still makes a really great point. So this is a correlation analysis that was published back in 2013 and looked across studies. It looked across human studies in a few different areas of injury, so burn, trauma, and endotoxemia. And then it did that in mouse models. And it looked for correlation amongst the human models and amongst the mouse models and then human to mouse. And the conclusions from this were that there's very little correlation between human and mouse data. And there's some correlation within human data in certain disease areas and less so in others. And there's a very poor correlation between two or more mouse models. And there's many things that drive this. A lot of it is what is measured in the study and how it is measured when it's published, because this was a retrospective analysis of published studies. The other challenge is that many of the mouse models and a lot of the poor correlation there is because people are doing things in inbred mouse models get further and further away from the human situation. And I would also say that there's a tremendous amount of data showing that there's poor correlation between some of the well-established cell lines. So things like HeLa cells, MC3T3s, HepG2s, and in vivo outcomes. And a great example of where primary human cells have really stepped up and provided better translational information would be the example of using primary human hepatocytes in ADME studies versus using something like HepG2. And so this is, I think we are accepting this probably amongst this group of people to a high degree. And so we turn our attention now, so if we're going to work with human cells in culture as a surrogate for doing studies in live humans, where do we get that tissue? So there's a few different places that we source human tissue for the purpose of isolating cells. But I think it's important to understand those avenues and some of the complexities related to those. So the first place we get them is cadaveric tissue. So this would be a deceased non-heart beating donor after cardiac death. And in this case, these tissues were donated and consented by the donor or donor's family to be used in research. Very rarely, but sometimes these tissues are also going to be used in transplantation applications. And after the death, the tissues are collected and processed and cells are generated. Then if you look at the second category here, the heart beating donor, so this is probably the most typical situation for the big major organs that are used in transplant, so heart, liver, lung. The donation occurring after brain death, but before cardiac death. Organs are prioritized and covered for because that's the primary reason that people are kept on life support to do these types of things. And then
Sharon?
the non-trans, sorry?
Sorry, Sharon. It looks like you have a little bit of a bandwidth problem. So if you have other applications open, I would suggest you to close. I turn off your video, but I'm trying to, if you have other applications open on your computer, it might be a good thing to close it all.
Okay, I don't think I do.
Okay, okay.
I don't,
It's okay. It's just fluctuating a little bit. So I just wanted to let you know that. So
Okay, I'm so sorry. I don't
No,
think
it's
I have
okay.
anything else open.
it's fine. It's fine. Keep going. We'll work through it.
Okay. Okay. So again, from the heart beating donors, the organs and tissues that are collected have to be consented for research use. And then the third category, which is where birth tissue falls in, is the living donor. And here, the tissue is recovered from a donor that is still living. And this could be from elective surgery like lipoaspiration. It could be a pathology specimen. A tumor has been removed and there's excess tissue, and you're going to now use the rest of that tissue to isolate cells. Or it could be from the birth tissue after the live birth has happened and the tissue is going to be discarded. And the donor also has to provide consent in this situation. But this situation is very different than the previous two situations because the donor is still alive, and you have to, in the consent for the tissue, you have to consider all the information that you're going to gather while using the cells from that donor, and you have to make sure the consent is a little bit more robust in terms of protection. So just something to file in your mind as you go forward in this space. So there are very unique aspects of birth tissue as a primary cell source. First of all, unlike a cadaveric or heart beating donor that has died for reasons of disease or trauma, this is a very healthy, plentiful tissue. There are about 3.8 million live births in the United States each year. About 10 to 15% of these are donated today, and certainly the potential of that number to go up in the future. The tissue is collected after birth, so there's no risk to the mother or child. And the placenta is very unique. It's the only de novo organ that is formed in an adult from scratch after birth, which makes it very unique. And it's also chimeric, meaning that it is composed of cells that derive from the mother, but it's also composed of cells that derive from the baby. And so you have this opportunity to access germinal origin cells without controversy. You're not sampling the baby. You're not doing anything with fetal tissue. Everything is after the birth, but the origin of the cells within the tissue, in part, is germinal. And there are many relevant cell types in these tissues. So if we look now at the structure of the placenta, you see the baby in the sac, and hopefully you can see my cursor. This is the amniotic chorionic membrane, and over on this side of the placenta where the cord connects, this is a disc of tissue, a thicker disc of tissue. And if we blow this up, this side is the fetal side where the umbilical cord is attached, and these are the fetal blood vessels. And these little tissue extensions come down into the maternal blood sinuses, and they're called cotyledons, very plant-like. And these and the maternal blood now form the maternal fetal barrier. Then the decidual tissue on this side is maternal in origin and contains a lot of interesting cells. So I'm sure you guys have all heard of mesenchymal stem and stromal cells, so MSCs. You can isolate these from any part of the placenta. They can come from the umbilical cord, they can come from the maternal side or the fetal side. From the umbilical cord, we also get the large vessel endothelial cells, which are widely used in research, and smooth muscle cells. And from the maternal side, in addition to the MSCs, we can get small specialized endothelial cells that form these spiral arteries that empty into the blood sinuses. And there's also maternal macrophages that help fight infectious diseases and deal with removal of debris, et cetera, from the tissue during development. And on the fetal side, we have some populations of placental stem cells that are fairly unique. And I'm going to talk about those a little bit later. We also have the specialized endothelial cells on the fetal side of the tissue. fetal macrophages, which are called Hofbauer cells, and the trophoblast, which line these cotyledon structures and form the first layer of the barrier against the maternal blood and control what passes. So the MSCs, probably, again, everyone has heard of these cells. This is what they look like in culture. They are derived from the connective tissues, so cord, either side of the placenta. They can also be derived from adult tissue. Fat and bone marrow are probably. They have a very definitive marker profile, but a lot of people will always say about MSCs, they're the things that stick to plastic when you first start a culture, and then later you can see expression of these markers, but none of them allow the cells to be separated by positive selection. They have a multi-lineage differentiation potential, so they can be turned into bone, they can be turned into fat, they can be turned into cartilage, and potentially some other tissue types as well. They're very actively used in regenerative medicine and immunomodulatory therapy. And I thought I would just pop this up, because we are still very much amidst the COVID-19 pandemic, and the FDA has approved a 60-patient study wherein the cells, MSCs from the umbilical cord tissue, are going to be tested for their ability to mitigate the cytokine storm in patients with advanced respiratory distress syndrome who have COVID. And so now let's talk a little bit about the stem cells that are within the placenta. So these originate on the fetal side. They can be isolated at any point in gestation. They can be found in the amniotic fluid, the chorionic villus, or the fetal portion of the placenta. We work with them solely after birth, so we get them from the placenta after a term birth. And they are enriched after isolation by positive selection with a marker called c-KIT. They share some markers with MSCs, but they also have some unique markers like CD17 and SSEA4. They also have a multi-lineage differentiation potential and a highly stable phenotype and karyotype. Chromosomes are very stable, long number of passages. These are also frequently applied to regenerative medicine, being advanced in autoimmune diseases and things like necrotizing enterocolitis. And we know from looking at a male, if a male baby is born, we can go back and do karyotype analysis on the isolated cells and show that they are a male origin. You see the little Y chromosome here. And so this lets us know that they are germinal origin versus maternal origin. And then there's the maternal fetal barrier, which we mentioned a little bit earlier, but this is a really important part of the placenta and the job of the placenta. It is formed by trophoblasts. So if you can see my little cursor over here, there are progenitor cells that form this syncytial layer of trophoblasts that have fused together to make a giant syncytia. And so this is in immediate approximation to the intervillous space where the maternal blood lies. And then underneath that is a small layer of basement membrane in the fetal capillaries, and then this is the fetal blood. So this structure right here forms the barrier. And you can imagine why it would be very compelling to model something like this in vitro, and especially have it be human. Now, to date, most of the attempts to do this have used a choriocarcinoma cell line called BWO in place of the trophoblasts, and HUVEC in place of the fetal capillary endothelial cells. And while that begins to get us there, there are a lot of features missing from those by not using the actual cognate cells that form that in vivo. And the barrier is going to control transport of drugs and toxicants, whether or not that happens across the maternal fetal barrier, but also just the exchange of waste and nutrients. And most of these things happen through active transport mechanisms across the capillaries and the trophoblasts. So these are highly specialized cells that do this particular job in the placenta. And then the umbilical vein endothelial cells, otherwise called HUVEC, you've probably heard of those before, and these are probably the most widely used cell that derives from birth tissue used in research. And they're used widely for angiogenesis assays, tube formation, basically, and vascular toxicity studies. And they're very favored in regenerative medicine as well because they're easy to access. The tissue is plentiful, They have very robust growth kinetics and angiogenesis capacity. These cells as well, so this happens to be an example from a male child that was born. And again, we have the karyotype, and we look and see that the endothelial cells lining the umbilical cord are indeed fetal in origin, they're male. And there's mixed literature on this in the literature. The opinion on this is mixed, whether these are maternal in origin or fetal in origin, but our data to date suggests that they're fetal. So if we look at the HUVEC in vitro, these vascular cells express things you would expect them to express: CD31, CD106, VEGF receptor, and some other markers. They're also negative for CD90 and CD45. They're very migratory cells, and they're capable of tube formation in standard assays that are used to measure that type of thing. They have prototypical cytotoxic responses to things that should be toxic to endothelial cells. Examples there are doxorubicin, lipopolysaccharide, LPS, which is an inflammatory peptide, and silica nanoparticles. These particles are often used in diagnostic sensors, and they're known to have some adverse effects on endothelial cells. And I'm going to show you a couple of examples of how we use those cells in vitro. But before, I want to talk just a minute about the role of oxygen tension in development and homeostasis, and how that can impact the measurements that we take in vitro with these cells. So as we are all standing and sitting today, the air that surrounds us is at about 21% oxygen saturation. So that's 160 millimeters mercury, and that's pretty high. If we were to look inside any tissue in our body, except for possibly the lining of our mouth and nose, we would find oxygen concentrations, oxygen tensions much lower, generally between 5 and 10%. And in embryogenesis, that is a particularly low oxygen environment. This promotes proliferation with some limited differentiation. And the gradients of oxygen that form control morphogenesis to a great degree. It's important to recognize when we take measurements in vitro, the physiologic responses that we get from the cells are going to be governed in part by the environment that we have them in. And this would include things like oxygen tension. It seems like a very simple thing, but it can actually have an impact. And as we take the measurements in vitro, everything we see is going to be a combination of, what are the inherent properties of the cells and what is the contribution of the culture environment that we've placed them in. So when a tissue comes in to the lab, one of the things that we do actively is so this is hopefully nobody's too squeamish, but this is a beautiful placenta on the left here. And we generate a cell suspension from this to begin to isolate all the different cell types. And from that, we parallel process and establish and make basically a lot of cells that is at our standard conditions. So if you have an incubator in your lab and it's hooked up to CO2 and that's it, it's going to be running at the 21% oxygen that we're breathing in the room. And if you pipe in nitrogen or take some additional measures, there's several different systems to get you there. You can lower that down to a physiologically relevant oxygen tension, or how we abbreviate that is PRO. And so when we are isolating cells from the same lot, same placenta, same pool of cells, we're parallel processing them into the standard atmospheric lab conditions, and we're also processing them into a lower oxygen tension, which is intended to be more physiologically relevant. And then we begin to observe how they behave in different situations. So one of the first things we notice is that, by the time we reach the second passage in culture, the number of population doublings we can achieve is actually higher across multiple lots in the lower oxygen tension. And we do see the cells are a little more lively. They may be a little bit smaller in size. So there's some signs, I would say, that the higher oxygen might make cells a little more senescent. they all profile like endothelial cells. All the standard markers are there across these lots. But the thing that popped up that was interesting to us is that expression of slug, which is associated with a more progenitor-like phenotype, was significantly higher in the lower oxygen tension. And so that was kind of a clue that things were not fully equivalent. So we moved on to look how the HUVEC behave to known toxicants when they are basically born and raised in the 21% oxygen conditions, which are the red on these graphs, or whether we are keeping them at physiologically relevant oxygen conditions, which is the blue on the graph. And you see a pretty standard doxorubicin concentration response curve here. Nothing surprising about that. You would expect it to be toxic, and these IC50s are within range of what you would expect. The thing that was interesting is that whether we were looking at IL-8 production, which becomes a surrogate for protein synthesis with doxorubicin, or whether you were looking at cellular energy levels, so ATP, in both cases, the IC50 was shifted to the left to a lower concentration for the atmospheric oxygen set of cells. And what this suggests to us is that maintaining cells at an atmospheric oxygen level causes them to be a little more susceptible to damaging agents. And this is something that we saw also. This is another example, just looking at the silica nanoparticles, and we exposed the cells to 3 different sizes of nanoparticles, the 20 nanometer ones, 80 and 100, over a course of 24 hours, and took a few measurements. So ATP generally goes down with toxicity, LDH goes up with toxicity, IL-8 is an inflammatory molecule, and if you look at the heat map, you can see that we begin to see some patterns of damage, possibly some differentiation between the high and low oxygen, but the one that was probably the most interesting was the 100 nanometer nanoparticles. And here what we're seeing is the same type of pattern we saw with doxorubicin. So if you look at the red dots here, you see a sensitivity to the silica nanoparticles at lower concentrations than we're appreciating in the physiologically relevant oxygen. And mirroring that is an increase in LDH. Again, a shift to susceptibility when the oxygen conditions are atmospheric versus physiologic. And so the way this could play out is that you would wrongly predict what an LD50 might be for drugs like these, reagents like these. And so, for us, this is just a very neat example of how cells that are start exactly the same can become a little bit different just simply by the way that you're handling them, culturing them, and assaying them. What we are working on now is profiling the phenotype of these cells and their functional performance and tox responses across multiple donor lots and then multiple cell types out of the placenta as well. We're doing some crossover studies to clarify the role of oxygen tension during the isolation phase, the culture phase, and the assay phase, because it's important to understand where the differences are coming in. And ultimately, our goal is to build a database that includes the donor attributes and all of this performance data so that we can better understand how the models that we build are impacted by those elements in the answers that we get, and how we can better select cell lots or types for specific applications. So just to summarize what we've talked about here, so birth tissue, again, is a very plentiful, rich source of cells. It's relevant to preclinical testing as well as clinical applications. You can get multiple cell types from a single donor, including vascular cells, macrophages, stem and progenitor cells of multiple varieties, and trophoblasts. A lot of these can go on to do things like maternal fetal barrier modeling. The primary cell biological responses are also influenced by not only the type of cell they are, but the culture environment, and that's particularly important for in vitro and vivo translation. And this would include things like what cell types are present, the configuration of those in 2D versus 3D, which we've heard a lot about during the course of the meeting already, and the milieu that cells are placed in. So the media, the oxygenation, physical forces, and things like that. So a lot to think about. But I hope that you will consider birth tissue as a potential resource for primary cells as you go forward and do the work that you do. And I'd just like to thank the organizers for including us and being part of this really interesting program. If you have questions and don't want to ask them now, there's an email address here where you can send us a note, and we hope to hear from you. Thank you very much.
Sharon, thank you for that excellent presentation. This is Marty. There is time for perhaps a question or two now rather than later. So let me begin. There are two questions. I'll at least take one. I think this can be generalized, but the person says, hello, I live in France and not all hospitals can organize to collect birth tissue. Any idea whom to ask when a mother wants to donate, etc.? So the logistics of getting started with this, presumably hospitals are already set up to a certain extent to do this. How do you get into that?
Yeah, so, and I would imagine in France, there's probably already some level of banking that is being done of cord blood, you know, which is a pretty popular thing to do. People will bank the cord blood either for allogeneic use in a public bank, or they'll bank it for autologous use in the future. People do choose to do that with their baby's cord. And those types of organizations usually represent an avenue in to have that dialogue. Because if they're donating the cord blood, what are they doing with the rest of the tissue? Would they sign a consent to allow that to be donated? And I would recommend that pathway if you're in an area where there are no tissue procurement. We have a partner locally here who procures, and they were able to go in and kind of tell our story and what we're doing at the foundation and our need for a different type of consent. And so they manage that on our behalf. Otherwise, you would have to have a coordinator that went into the hospital, got an IRB, and started a collection program on your own, or you could partner with someone who's banking cord blood. That's probably the fastest path.
And thank you for that. Perhaps one more question. How readily can maternal and fetal cells be separated? And how is that done in the case of a female fetus?
Yeah, so in the case of a female baby, you have a limited ability to QC at the end your cells and prove that they were fetal. You can do there's some genomic tests that you could do, you know, to prove that, but that would require you to have a concurrent sample definitively from the baby and the mother, which gets complicated. There are macro dissection techniques that are fairly reliable. And I think we have, for example, the placental stem cells, we feel pretty confident that we can macro dissect and get what we need. But the lowest risk situation there is to work primarily with male birth. But you really want to have ultimately a population or cohorts of cells that spread both genders. So, you know, that's not as ideal, but you can do some genotyping to prove origin.
Thank you. I think we'll move on from that and resume questioning in the Q&A later on. Thanks so much.
Great.